Hello students, this is Faizan Mirza. We are discussing the topic of enzymes at the moment. Enzymes, as you know, the features of enzymes you must have already discussed previously as well, that enzymes are globular proteins. They are made from polypeptides, which are chains of amino acids joined together through peptide bonds and various other bonds, which includes the hydrogen bond, the ionic bond, the disulfide links, and the hydrophobic interactions that help the enzyme attain a final shape. Enzymes have an active site, and into this active site, the substrate can fit in. And this is done by the lock and key hypothesis, where the lock and the key they fit in together. And the induced fit model also can be discussed here. Induced fit model states that the fitting of the substrate into the active site of the enzyme causes the active site to alter its shape slightly so that the enzyme and the substrate could be held more close together. So catalysis could occur. Apart from these, the enzymes, you know that they lower the activation energy by forming temporary bonds with the substrate. They are able to break the bonds and make the bonds, new bonds there. Enzymes, they speed up the chemical reactions. They are biological catalysts. Activity of enzymes is affected by mainly five conditions. Number one, the concentration of the substrate being changed, concentration of enzyme being changed, temperature being changed, pH being changed, and the presence of inhibitors. Enzymes, they convert substrate to product without themselves being changed in the course of reaction, and they are regenerated towards the end. So minute quantity of enzyme is available for catalyzing a large quantity of substrate into product. Enzymes are made using the chains from using the codes from the DNA, which turns the codons of the DNA into polypeptides, and the R groups of the amino acids, they are important, or whether they are present on the active site or elsewhere on the enzyme, because these are the ones which are involved in catalysis as well as the formation of those temporary bonds. The R groups on the surface of the amino acids of the amino acids on the surface of the enzyme, they are also immensely important because they are polar, and they make the enzyme soluble in the aqueous environment. Since all the enzymes, they are hydrolases, they need aqueous environment to work. Enzymes are made on ribosomes. Since they are proteins, so they are made on ribosomes or on rough ER. So this sums up the basic features of enzymes carry out anabolism as well as catabolism, which means the metabolic reactions. These metabolic reactions could either be the ones which require energy, for example, the endergonic reactions in which the energy is used up, and the catabolic reaction where energy is released, the exergonic reactions where the substances, much simpler substances, are made. For example, if you take an example here, you take this substrate of hydrogen peroxide here, and you add catalase enzyme. The bubbles of oxygen, this is the reaction that will be catalyzed. H2O2 will be converted into H2O and oxygen through the enzyme catalase. You added catalase here, 10 volumes of substrate is added here. The catalase 1 ml is added here. This is released gradually, and then the oxygen which is released is collected here. You can count the number of bubbles, and you can note down the volume in the measuring cylinder, and you can note the gas collected per 30 seconds. And with this, you can plot this graph where the time is taken on the x-axis. On the y-axis, you have the volume of oxygen produced, and you can see that the graph starts to rise steeply, and then it starts to become parallel to the x-axis. The rate of reaction is fastest at the initial rate at the initial time, which is the initial most moments of the chemical reaction, because all the active sites are vacant at the moment. To calculate the initial rate of reaction, we just draw a gradient at 30 seconds, and we calculate the slope of the curve there, and that tells you the initial rate of reaction at that time. This is what I've just discussed here. You just plot a graph of time against volume of the product per cm cube at 30 seconds. So you just find the gradient of the curve. This gives you the initial rate of reaction. And you can just see this from the graph as well. The rate of reaction is fastest in the initial few moments of the graph. That means that even before 30 seconds, 10 seconds, 15 seconds, it's even more faster there. Why? Because all the active sites are vacant, and the substrate can easily fit into all the active sites which are lying free at that time. So as the course of reaction proceeds, as the time goes on, the active site becomes 100% saturated or nearly 100% saturated with the substrate. So the rate of reaction, which is the rate of formation of product, it starts to gradually decrease, and it starts to eventually becomes constant. So the rate of reaction actually tells you how quickly the products are being formed. So rate of reaction could either be followed by either the rate of disappearance of the reactant or the rate of appearance of the product, These are the two ways in which the rate of reaction could be followed. For example, you take an example of starch being converted into glucose. Starch is a substrate. Enzyme amylase can turn it into maltose and then into glucose. It can be broken down. The glucose is the final product. Starch, which is the reactant, you can just follow either the disappearance of reactant over time, or you can just follow the appearance of the product glucose over time. So what do? You just take a cavity plate or a white tile, and on a white tile you just mark these with 2 second in 2 minutes intervals here, with a marker here, and you have a stop clock as well. You take a beaker, and this beaker, you add water. You make a water bath here. You add your starch suspension here, and to this starch suspension you will add amylase, a known quantity of starch, a known quantity of amylase. When they are added, you add a glass rod to it. You start the timer at 0 minutes. You take out the drop from the glass rod and you place it here at the spot where you mentioned 0 minutes. And when you place it here, you add iodine and potassium iodide solution onto that spot. The spot will turn blue black, telling you that at 0 minutes starch was present. And then you let the stop clock grow from 0 minute to 2 minute to 4 to 6 to 8. And at every 2 minutes, you just keep on taking out a drop from here, place it on the white tile, and immediately add iodine and potassium iodide solution. So that will actually tell you that how the rate of reaction is actually, the color actually starts to discharge. Initial few minutes you will see that there will be a strong blue black color here, but as the time progresses you will see that the color starts to discharge, and towards the for example 14 minutes or 16 minutes you will see a much lesser blue black color, and towards the end of 22 minutes you will probably be seeing that the reaction have just stopped. So you know that the reaction is completed. So rate of disappearance of the reactant has been followed. The rate of disappearance of reactant, you know the time, how much time it took for this reactant to disappear, and this can be turned into rate by dividing one with the time, which was total time, and this can be repeated at various conditions. For example, at a selected temperature, or using certain kind of pH buffers, or any other condition that you want to just follow in this reaction. This reaction can be put in those conditions. Along the temperature axis, if you take temperature on the x-axis and rate of reaction on the y-axis, you know that at a very low temperature, the rate of reaction is very slow. The enzyme is deactivated. Deactivated means that it will just not collide with the substrate, and since no substrate enzyme collision occurs, the rate of reaction will be very, very, very slow. As you start to increase the temperature, for every 10-degree rise, there will be about doubling of the rate of reaction. At about 40 degrees, you have the optimum temperature of the enzyme for human enzymes, and this is slightly higher than the body temperature. This is to accommodate for fever, and if this is the point where you have maximum enzyme substrate complex forming, because the active site is intact and the substrate is colliding very well with the enzyme. The enzyme is also colliding right in an appropriate way, and the rate of reaction will be very fast. The product turnover will be very fast. But if you go beyond this optimum temperature, the rate of reaction will start to decline. It doesn't mean that the collisions are not occurring. Since the temperature is increasing, the collisions will definitely be occurring at a very fast pace, but the enzyme will become denatured. The 3D shape will be distorted. The active site will not be functional there, because as you increase the temperature, the hydrogen bonds, they start to break. When the hydrogen bonds, they start to break, they are breaking, and when their hydrogen bonds are breaking, you know that the three-dimensional shape of the enzyme will be changed, and the active site will not accommodate the substrate anymore. When we compare the effect of pH on temperature, you can see at different pHs the effect of pH on the rate of reaction is also varying. It depends on which enzyme we are talking about. pH changes, extreme changes in the pH, they basically break the ionic bonds, not the hydrogen bond. The ionic bond between the R groups, and the R groups group interaction are important in the tertiary as well, the quaternary structure. So once the enzyme is distorted, the active site is distorted, the ES complex cannot form, and the rate of product formation starts to decrease. Most of the human enzymes, they work best on the alkaline medium. You can see these three graphs here, the optimum pH for this enzyme and these two enzymes. You can see how the range of pH is different for each enzyme. The range basically tells you the range over which the enzyme is still functional, and the optimum of each of these is also slightly changed. So if you increase the enzyme concentration or substrate concentration, they both follow the same kind of a graph. You increase the enzyme concentration, the rate of reaction is very fast initially, and then the rate of reaction starts to slow down. If you are taking enzyme concentration on the x-axis, then eventually the substrate concentration will become the limiting factor. And if you are taking the other kind of graph where you take the substrate concentration here, you will see that the enzyme concentration will become the limiting factor. The substrate concentration when taken on the x-axis, you can see this graph when you're going from low to high. This is the initial rate of reaction which you are plotting on the x-axis, on the y-axis. So the graph increases steeply. The maximum point which the graph reaches the Vmax. That's the point where the maximum rate of reaction will be present there. The 100% active site will be occupied at Vmax. This basically is the maximum turnover of product formation. So that's the maximum rate at which the enzyme can function. This is Vmax. This can be seen on the y-axis. You can just plot this graph and you can find out the value, read of the value from here. This is Vmax. The 100% enzyme is functional, 100% efficiency of enzyme, and 100% active sites are occupied by the substrate. If you just divide this by 2, this is half Vmax. 50% of the enzyme efficiency, 50% of these active sites are saturated with the substrate. If you just extrapolate this and you touch the graph and you drop down the value on the x-axis, the substrate concentration here is at which the enzyme becomes 50% saturated or came at 50% efficiency. This is called as the Km, or the Michaelis-Menten constant. This Michaelis-Menten constant here shows to you that at a very low substrate concentration, enzyme was 50% saturated. So this Michaelis-Menten constant actually is used to predict the affinity of the enzyme for its substrate. If the affinity is high, the Km would be low. If the affinity is low, the Km would be high. If the graph is given to you, then you just take the value of Vmax from where the graph is parallel. You can see this on the grid that the graph is actually parallel from the axis, but in reality it's not so. The graph never becomes parallel to the x-axis. We cannot follow that in a chemical reaction. So we if it's not given on a grid but on it's sketched in this way, so you can see that the Vmax is plotted slightly higher where the graph ends. So if it's given on this kind of a plot, or if it's given on this kind of a grid, the Vmax can be plotted accordingly. Affinity, as I told you, that the increased affinity is for enzyme and substrate, the Km value would be decreased, and if the Km value is high, the affinity of enzyme substrate complex formation would be low. And this is just the maximum rate of formation of product, which is Vmax. So Vmax and Km, they both are different conditions. They both are independent of each other. You can't use Vmax to predict or Km, or you can't use Km to predict Vmax. They both are irrespective of each other. The next that we are discussing is enzyme inhibition. Enzymes can be inhibited using inhibitors. If the inhibitor occupies the active site, this is the active site, that the substrate occupies the active site. But if the this inhibitor, it occupies the active site, it will occupy the active site, will prevent the substrate from coming there. This causes the competitive inhibition. Non-competitive inhibition, in this the substrate can still attach to the active site, but in the presence of non-competitive inhibitor, it will bind to any other place on the enzyme, which is called as the allosteric site, will form bonds there. Forming bonds here, this distorts the active site, and once the active site is distorted, the substrate cannot fit in. This is called as a non-competitive inhibition. So you can see that the competitive inhibitor does not need to have the exact same shape as the substrate. It can have a different shape, but it will form bond with the active site. The active site becomes occupied. The allosteric site becomes occupied during the non-competitive inhibitor, and the active site shape changes slightly, so the substrate cannot be accommodated there. You can see the graphs here, how the without inhibitor, how the graph goes very high. This initial rate of reaction versus substrate concentration, and how the rate of reaction decreases when you are using a competitive inhibitor or a non-competitive inhibitor either. So when we are discussing these three graphs, so I'm just using the key here, the blue, purple and yellow color to denote this without inhibition, competitive inhibition, and non-competitive inhibitor. You can see that in the normal graph, you can see the Vmax and the half Vmax, and you can see the value of Km as well. So if you just plot it here, you see that the Vmax is reached in the same way in both the graphs. That's without inhibitor and with the competitive inhibitor. And the half Vmax is also the same. But if you extrapolate this half Vmax value and you touch both the graphs and you drop these down, you will see two different kind of Km values. At the Km was way lower when the inhibitor was absent, but in the presence of a competitive inhibitor, Km was increased. So an increased Km means that the affinity of the substrate for the enzyme got changed because of the presence of the inhibitor that occupied the active site. The Vmax here in the non-competitive changes altogether, so it will have a different half Vmax. For this half Vmax, the Km value would be same. So the Km value will be same for this kind of graph here. So the Km stays the same, the Vmax changes. So this is one what condition we study, that in competitive inhibition Vmax stays the same and the Km changes, where in the non-competitive inhibitor the Vmax change and Km stays constant. Lastly, we are discussing here the end product inhibition, the last part of inhibition actually. Then there are various series of reaction in which the end product is the one which we use. This is the substrate which is turned into product 1 using enzyme 1. Product 1 turns to product 2 using enzyme 2. Product 2 turns to product 3 using enzyme 3. The product 3, the end product goes and inhibits the first enzyme or any enzyme in the cascade. So in this case, it went on to inhibit the first enzyme. So when this first enzyme got inhibited, the first enzyme gets inhibited, it cannot attach to the substrate anymore, and the product 1 formation will start to decrease, and the product 2 will not form and product 3 will not form. And this is how the product 3 controlled its rate of formation. So irrespective of how much substrate you have, the product 3 will not be made in that huge quantity. So as the cell ends up using all this product 3, even the one that was attached to the enzyme 1 that it was attached with while inhibiting it. So once this product 3 has been used up, the entire loop would start to function once again, since the inhibition of this enzyme would end, and the substrate will then attach to the enzyme 1, and the cascade of events can then continue. The last thing that we have to discuss here is how the enzyme can become immobilized. We'll take an example of people who are lactose intolerant. They cannot digest lactose because they don't have the enzyme lactase there. So enzyme, what you can do to help them, you just mix enzyme with the alginate, you stir it thoroughly, it forms a uniform mixture. You add it in a syringe and drop by drop pour it in calcium chloride solution. Beads of the trapped enzyme will form, and these can be washed using distilled water. This removes excess calcium chloride and the unimmobilized enzyme. You fill a glass column with these beads and you pour the substrate and you collect the enzyme-free product in the beaker. This is how lactose-free milk is produced. So this is the column that you can see that that we'll be filling with this enzyme. And when you fill this column with the enzyme, the beads here, and you add the substrate from the top, these as the milk just goes on from this column, these are the immobilized enzyme. The lactose in this milk will be broken down, and the lactose-free milk can be collected. This will be glucose and galactose here. So this is the basic function of how enzyme-free product can be made using this immobilizing technique using sodium alginate. That's it for now. Thank you so much.